the AIDS HIV lecture today. Continuation with what we said yesterday. Again, you can see from this gene segment of HIV, and this happens to be, I just stopped yesterday on this slide, and this is a gene segment of HIV, and then if you pay attention to abbreviations in the end, LTR stands for long terminal repeat, POL, gene segment, means polymerase, ENV means envelope, VIF means viral infectivity factor, VPR means viral protein R, TAT means transcriptional activator, REF means regulator of viral gene expression, VPO means viral protein U, and NEF means negative effector. All these jargon basically is for good use because you need to know how is it that this virus is causing problem. And say it again. No, we've already announced that attendance is on. So attendance is on, is from 11 to 11:05. It's going to remain on for the next 2 weeks. And same goes for all these things. Okay. Now, I want you to pay attention to, I'm going to take each one of them separately. So you can see from here, there's a functional element of each and every gene. For example, we talk of LTR, which is a long terminal repeat. That's a transcription of viral genome. It is the integration of viral DNA into host cell genome. So this is the binding site for host transcription factor. So this is like in the end, right? I'm going to cut it half, hold it from the side, and put it into the genome. So this is like my two hands. I'm going to break it apart and cut it up. So these are long terminal repeats. Then we have GAG. You can see from here, GAG is a nucleocapsid core and matrix protein, and we did discuss the other day. I gave you all the important proteins and what are their function, but this is the gene sequence for that. For POL, which means reverse transcriptase, protease, and integrase. These were the three important enzymes I told you the other day. What I did not tell you at that time was ribonuclease. So ribonuclease is also part of that. Viral core proteins. Remember, I said there are two important core proteins which are exhibited on the outside. Like a globular one was GP120 and GP41. And then we have VIF, which overcomes inhibitory effect of host cell enzymes. So remember, this virus is so clever that even your system wants to fight it off. It comes in its genome, a gene for that will overcome the inhibitory effect of host cell enzyme and eventually will promote replication. So you can see these genes are the targets of the anti-HIV drugs. Okay, because the idea is we want to stop replication of the this particular virus. Then we have VPR, and VPR increases viral replication. So of course, it's going to increase replication, and we have HIV infection of the macrophages. Required for elongation of viral transcript, promotes nuclear export of incompletely sliced viral RNA. Now, remember I said yesterday that we'll just have you answer 1 point extra credit during the lecture. So be prepared for that. Now, this particular VPU over here, it downregulates the host cell CD4 expression. Okay? What do you think? Why the virus wants to down-regulate host cell CD4 expression? My first question that, again, the rules would, why? Why is it the virus wants to down-regulate CD4 expression? Yeah. There's no maybe. Be particular. T cell won't be expressed. No. Who wants to get, yeah. Decrease the immune. Be specific. I mean, we already done immunology. I don't want you to give me like maybe, sort of. No, that's it. You took your time. Who else? Yeah. Good. So you get 1 point. T cell don't recognize the virus because virus has to be recognized. Okay, virus has to be recognized. So CD4 receptor is going to see the virus. So the virus doesn't want to be recognized, and so you can give your name to Ransford for 1 point. But that's all for today. You're done for today. You can go. Okay. So so that works for CD4. Okay. Then again, you say CD4 regulates host cell CD4 and class 1 MSC expression, enhancing the release. What is the relationship with CD4 expression and class 1 MSC receptor expression? Why they are tied together? That this particular negative factor is going to downregulate. Part of academic integrity, I don't want you to Google the answer. What was the question? Okay. Well, if you keep on asking me, that delays my lecture. But what I said, right, what is the link between host cell CD4 and class 1 MSC expression? Why would this gene want to downregulate it? So April, you'll use 1 point if you're going to prep others to do that. So negative for if you're going to help others to do that. Yeah. Louder. So what's the relationship with CD4 and MSC class 1? What is the relationship between these two? Why would the virus go for that? So this is going to be minus point for you because CD4 does not recognize MSC class 1. CD4 recognizes MSC class 2. Okay, so we'll start with the negative marking as well. We go back to your immunology test and take 1 point away. Okay, anybody else want to say something? Okay, let's move on. And I'll explain that to you if you don't know. Anyway, well, I said I'm going to explain that to you. Okay, now I just showed you HIV genome. And keep in mind that if you look, I mean, I cannot show you both pictures at the same time. The linear gene that you saw as indicated different color blocks. What are important thing other than the function of genes that I've given over there? Some gene use same sequence as other gene. So you may see the sequence, if you look at go at their amino acid sequence, you will see is same. But what happens is that we have this host cell RNA polymerase that may read it differently because of this overlapping thing. So it is like A, B, C, D, E, F. So maybe it's going to pick up ABC, it's going to pick D, E, F. So reading will be different is in terms of the enzyme sequence. The other important thing is that if you go back to the same, you'll see some of them are shaded blocks. So these shaded blocks, which are separated by lines, indicate genes whose coding sequence are separated in genome and require RNA splicing to produce functional mRNA. So that again is a concept for the basic genetic is that you have a gene and then this enzyme is going to slice a part of that gene and insert it. Okay, that again, we have very specific enzymes that are going to take a bigger chunk of a gene and slice that portion. For example, let me give you an example if I could. So you can see from here, some of them are, this is separate. So you can take the LTR separately, just cut it up from here. But if you were to cut GAG, so you have no way of cutting GAG, and you're going to get a portion of PO as well. So these are like overlapping sequences. You can still do that, but the RNA polymerase is going to read it differently. That's what they're trying to say. So for overlapping genes that we normally see in this case is where it is going to cut, because these genes are going to lead to proteins, transcription and translation of proteins. And these transcription and translation of proteins are going to act as enzyme, because enzymes are mostly protein. So that's what they're trying to see in terms of overlap, where the things are for some of the important genes that she. But both of them, as I said, is a long segment, and you want to break it up from both sides, put it together, insert it into human genome. All right, now pathogenesis. Pathogenesis of AIDS or HIV. So you can see from here, let me enlarge that for those of you who want to understand it. So you can see from here, this is a virus, okay? And virus basically is going to get attached to CD4, right? CD4. So what is CD4? What is it? What exactly is CD4? Huh? It's a receptor. It's a receptor, right? The question is, where is this receptor expressed? Yeah. Louder. You have to be louder. Okay, go ahead. Surface, okay. Be specific, yeah. Which T cell? Incomplete answer. Okay, there are two types of helper T cell. You have to be very specific. There are Th1, Th2, Th3. Which one? Exactly, give me a very specific answer. Yeah. Louder. Don't get excited. Okay, okay. One at a time. I cannot share it to all of you. Okay, let's turn towards this side. This hard spot over here, these people are silent. Go ahead. Anybody want to say something from here? Oh, you're worried about negative. Okay. CD4 receptor is present on quite a few cells. One of the cells that has it is CD4 T cell. CD4 T cell itself has a CD4 receptor. Okay. And what are the, it says over here actually, the answer is here. Monocytes have CD4s. Macrophages have CD4 and dendritic cells have CD4 on top of all the TH. It's not one TH1, TH2. All the T helper C cells may have CD4 T cell. But 1, 2, 3, 4. There are 4 different types of cells that can recognize HIV virus. Okay? Now, what happens after that, once they recognize that, they are going to go into functional change. And I'm going to go and teach you what we already discussed that. And most of them will go into different types of TH cells. And they are going to do what we talked about, immune response. Again, a typical immune response. And by the end of the day, that immune response will depress the immune response to HIV. And the reason is because HIV wants to do the damage in the presence of immunosuppression because it's acquired immunodeficiency syndrome virus. Now, the other scenario is that but when I taught you immune response to viruses, what is the hallmark of immune response to viruses? What is the hallmark of immune response to viruses? Okay, I'll take this negative point away. Yeah, go ahead. People get scared. Yeah. Say it again. Very general. This is not. Hallmark mean very specific. Hallmark is a key. Yeah. Very good. You got it. CD8 T cell. CD8 T cell is the hallmark of viral infection. Okay, but why are we talking about CD4? Why are we talking about CD4 in HIV? Is that the point? That is one of the problems we have with HIV. Normally, CD4 T cells are good for bacterial infections. Okay? In this case, CD8 T cell should initiate a good response. So what this T cell does, this virus does it, is not going to go for his natural immune response. It's going to take alternative pathway and make friends with CD4, because CD4 are not the professional cells to deal with this kind of viruses, right? They are good for bacterial one. They can't even theoretically present it, because the presentation of viral proteins is by CD8 T cells. So if you do get TC response, which is cytolytic T cell, the other name for the CD8 is, you get reduction of viremia and you make neutralizing antibodies. So that probably will show you that response. But what happens is, since the HIV virus infects all cells expressing CD4 cell, and that includes 4 types of cells: monocyte, macrophages, dendritic cells. What will be the outcome if these cells are killed? If all those cells expressing CD4 T cell receptors are eliminated or killed by a virus, what will be the outcome for that? All your turns are gone. Yeah. Anybody else? And we'll come back to you if nobody else answers yet. Okay. Okay, just raise your hand. No, I said I'm not. Say it loud. In the blood? What do you mean? No. Okay. Let me take him first. Yeah, go ahead.
Yeah, when all these, when they're all dead, monocyte, macrophages, dendritic cells,
yeah,
then the immune deficiency is opened for infection. That's why there's all of these other, how you say?
You want to share your points with her?
No.
If you're asking her, yeah. Just hold it.
What I was trying to say is that, the bacteria that we ordinarily would fight off will now become active.
Okay. So there's a term for that. Yeah. Say it again. Yeah, but there's a very specific answer for that. Yeah. Yeah. It doesn't need to recognize the virus. CD4 doesn't recognize the virus. Okay, all right. Let's take Ben, because nobody else has correct answer. Go ahead.
Doesn't the 120 receptor on the HIV bind to the CD4 so it can't infect those cells?
My question was, no, the thing is, if because of this virus, all those 4 types of the cell expressing CD4 receptor are killed, what is going to happen to the body? No, raise your hand and wait for it. Yeah. Go ahead.
Yeah, I'm saying that if all of them are killed, then the body's immune system is
Yeah, that's a generic thing. That's again a generic thing. Yeah. That's what he said. It's a consequence, but what something else needs to happen here. Okay. You want to say, Amanda?
I don't even know, but if the CD8 will also decline as macrophages.
This is a consequence. All right. She's go ahead. You may, if you're gonna push me for negative, you know. Louder. These are consequences. There will be no antigen presentation. These are antigen presenting cells. If you remove whatever you're telling me is correct, but by the end of the day, it is no antigen presentation. That makes sense? Because you remove antigen presentation. You are all right. But the technical term for that is you are killing antigen, you are removing them. Is that the point? So this is what's going to happen in terms that virus is going to persist, is going to disappear. Whatever you're telling me, if there's no antigen presentation, bacteria will not be presented. You have bacterial infection, fungi will not be presented, fungal infection. All these infections will come in HIV because HIV was to come and put up a fight with CD8 T cell. It doesn't want to do, so it's going to go and kill the antigen presenting cells, all of those cells. What was the cell, antigen presenting cell, which HIV normally, I use the word normally because I didn't teach you, has not killed so far? Which antigen presenting cell still remains even for HIV, after HIV? Yeah. I just told you 4 times. They're already gone. Dendritic cell. Yeah. B cell. You got it. B cell. B cell is an antigen presenting cell, and the virus did not eliminate that. Very good. I wish I could have given you 2, but 1. Very good. Very good. Okay, let's move on. Now, this is the route of infection that we've been talking about. And the funny part of that is, and I'm not going to discuss that, red one is a major route of infection in resource-rich countries. That's a very funny classification. I'm not going to go in detail. So the most important route of infection for resource-rich country is, again, heterosexual, homosexual, and male, as compared to resource-poor countries where it is heterosexual. Okay, not only heterosexual, but also vertical transmission. And there are a lot of reasons for that. I'm pretty sure if you were there the other day, I showed you this frontline picture, poverty, socioeconomic, lack of testing, and everything is part of that. But that actually is important in terms of our understanding of what is going on in terms of infections. Now, HIV replication cycle, as you can see from here, and how does it go into the cell, and what is going to happen is, because that's important. So the very first step, as we discussed other day, which is adhesion. So the virus is going to attach to CD4 receptor. Make sure you understand this concept. I'm not saying CD4 cell. I'm talking about CD4 receptor, which is present on 4 different types of cells. But most of the time, we think the target is CD4 T cell, and that's how we assess that. Now, we recently found out that it's not only CD4, so that's not enough for the virus to attach. It also binds with the chemokine receptor as well, because we're going to go for drugs for these 2. Okay, so it binds to these 2, and this binding is from GP120. If you remember the structure of the virus, GP120 was this globular attachment, blobs that are going to go and bind to that. So it's going to bring it into the cytoplasm, penetration, right, and form a vacuole. So you can see it's going to form a vacuole. The next step is when it kind of penetrates deeper, there's going to be fusion of viral envelope with the cell membrane. That fusion is going to be from the cylindrical portion of that glycoprotein, which is GP41. So GP120, GP41, very important in terms of the first step of virus attachment and penetration. And we will talk about the drugs which are there. Now it uncoats. Then you have reverse transcriptase. You remember the LTR? I showed the LTR with 2 LTRs on the side with the gene in the middle. So these are the 2 LTRs over here. It's going to take it apart, bind it, and insert it into the host DNA. So you can see your integration into host DNA. That's one of the important things is over here. So this is integration into host DNA. And then again, typical process, we talk about transcription, translation, viral proteins are made, nucleocapsids are made, virus is ready, it's going to pick up the viral envelope protein from outside, and then it's going to bud out and release. Okay, as we discussed that for most of these viruses that you saw in the past in terms of their replication, and all these are important in terms of what step are we going to take to stop all these kind of activities that are happening within the cell. Okay, now let's talk about the drugs. This is one of those viruses that I'm going to ask you for the drugs as well. As I said, that this is the only time we teach you HIV, AIDS, and you keep that in mind. And you can see from here, number 1 is that there's an HIV virus. There's virin binding to CD4 chemokine receptor. So this is a CD4 receptor. This is serpentine chemokine receptor. Both of them need to be attached and to be engaged before the virus comes into the cytoplasm. Okay? So this one is attachment absorption. The other one is a fusion. I just told you that the first step needs GP120 and the next step needs GP40. One. Good. It's going to uncoat. You have reverse transcriptase. It's going to do what it needs to do. It's going to go transcription, translation, and then all those products made different parts of the virus are going to be assembled together and wrapped up, and then you need to bud it out. So you need finally the expression of these proteins, and the virus becomes immature. Same picture that you saw in the previous slide. The only difference is that it's a little bit more detailed. I'm going to add drugs on top of that to give you an idea what is it that's happening. Okay? There you go. So you can see that naturally we have drugs. So I just put 4 lines of drugs. These are not the only one, but you can see if I was to stop that fusion of HIV membrane with the cell membrane where the viral genome is going to go into cytoplasm, I'm going to use a classification of gut fusion inhibitors. Fusion inhibitors. Okay. Then if I want to go and work on reverse transcriptase mediated, because reverse transcriptase or reverse RTIs are reverse transcriptase inhibitors. So we have reverse transcriptase inhibitors, another line of drugs which are there. Okay. Are you with us? Are you with us? I'm talking to you. Yeah. What did I say? I'm talking to you. What did I just say about the antiviral therapy? I want you to participate because these are important. You know, sometimes you come and ask from me, how do I do good in the exam, this and that. So some of the questions that I'm talking about, I'm going to ask you in the exam. So try to understand. And as I said, this is one of the most important lectures that you have in terms of therapy as well. Okay. So we talked about reverse transcriptase inhibitors. So they are important, very important. Then again, the other enzyme, remember I told you about the 4 enzymes: reverse transcriptase inhibitors, integrase, protease, and what was the 4th one? Ribonuclease. So these are 4 important enzymes which are there. So we can have integrase inhibitors over here, and then finally we can go for protease and maturation inhibitors, because we don't want expression of GP120 and GP41. What will be the advantage of protease in maturation inhibitors? What will be the advantage of protease maturation inhibitors? Well, let me focus on this side, because I've seen this is like hard spot, and these people are doing some other chores. I can tell by the participation. They're looking at me. Yeah. Yeah, you can. Yeah. I want you to participate. Yeah. What do you mean? You're not thinking? No. It's not a contribution. I just said it for you. I just want you to repeat what I said. Yeah. What is the advantage of that? Does not overgrow. Okay. Anybody else? You want to say something? Yeah. Say it again? Correct. But what exactly it means? Why do you want to use maturation inhibitors? Yeah, so last row. Yeah, last row on the left-hand side. You want to contribute? Not this side. Just hold on. Yeah. Remember, I asked the other day a question that, and there's a question to me as well, that if everything is recorded, everything's representing, attendance is not mandatory, why we are here, right? So I think the minimum expectation is that if you are kind enough to be here and giving your attention to me, I would want you to participate. So that's going to make best use of time. I don't think these chairs are good enough for you to prepare for the STAT exam tomorrow. I mean, don't worry about the attendance. Don't worry about the extra credit. Who cares? But try to participate. I mean, that's the whole point. I mean, I would want each one of you to think, because if you think, that will be good for you, so for your understanding. Okay. Now, in this case, what happens is that if you don't allow expression of GP120 or GP44, the viruses can never attach. And if they don't attach, they cannot replicate. Okay, so that's the reason we're going to use for these maturation inhibitors. Okay, you know. The clinical picture is based upon the pathogenesis that we just discussed earlier. And acute HIV infection, you get this symptom, very vague. The critical latency period is that, as I said earlier, of all the 4 cell types that I told you express CD4, why do we just look at CD4 T cell and not look at other cells in terms of finding out the extent of disease? The reason is because CD4 T cell, remember, are the major player. If you remember, when we were talking about the immune response, I said that they are the major players for response, CD4 T cell, everything comes. So if they're gonna go for the target cell, right, then again, we'll have the whole immune response jeopardized. If you remember, I said for bulls, it's like direct rows. That's what I said when I taught you the immune response. So this is a central pair. Then again, you just told me antigen presentation is lost. You are exposed to all kinds of opportunistic infections. And then if the genome is integrated into your host, it's going to cause all those cancers that are there, and it will also cause encephalopathy and wasting syndrome. And the picture is complicated, but the term that I use for most of viruses, latency, is going to go for latency. Now, if you want to look at the natural history of HIV infection, you can see that people may get infected without knowing. And if I was to kind of say something about whether a person has HIV or not as a physician, I'm going to look at the CD4 T cell count. If I find low CD4 T cell count, then I can start thinking of something is happening, because CD4 T cells are the major index for the natural history of HIV infection. And you can see all the way from normal to less than 200 per cubic centimeter is like a threshold. Then we are looking that this person is going to die. All the anti-HIV therapy is based upon maintaining a healthy CD4 T cell population so they can fight off infection. So you can see the moment we go to the decline, all those things will appear. Thrush, hairy leukoplakia, tuberculosis, pneumocystic catarrhine pneumonia, we talked about that. And all those, so to speak, mild infection that should not cause you disease, but it does cause you disease because you lose the main player. As you mean the main central player, and then you have, but it may take years. It may take years. Right? So one of the objective of the management of HIV patient is we delay that happening. Okay? And we want to increase the quality of life of the patient. So these are some of the opportunistic infections associated with HIV infection. So you can see number A is a hairy leukoplakia on the tongue. B is extensive oral candidiasis, which is a fungal infection. Number C is Kaposi's sarcoma over here. Number 4 are pneumocystis pneumonia. Number 5 is over here is cytomegalovirus retinitis. And then we have cryptosporidiosis. So these are classical signs very commonly asked, what are the opportunistic infections and tumors that normally HIV patients present with? This is like a terminal stage of the patients before they die. That, one thing that you will see over wasting of the muscle is there. You're going to lose the muscle mass, infections, and so on and so forth. Now, this slide is most important in terms of, I would say, I will just pick at least 5 different distractors from there for you to understand the concept over here. And I want you to, again, pay attention to the time frame that this virus can sit in your system for more than 10 years. Okay, without you knowing it, but you still can pass it to other people. So you're thinking about the years. Then you have CD4 T cell count. I told you, and the most important threshold is about 200, right? And then the other is viremia. This means HIV RNA copy. So once the virus is multiplicating, it's going to release those copies into the system, and we can take this up and determine the viral load. Right? And if you remember, I said viral load can be exhibited in the blood, but what other things can also give you an index of a viral load? What are the other things that you're going to look for other than blood to look for viral load? You have a lot of your turns. Let somebody else take that. Yeah. Anybody else? You have to speak up, otherwise I'm going to pick on you and ask you to do that. And this is something I already taught you. Yeah, Ariel. Okay. You see, there's a latency period. So delayed, okay. What was the question? I said that we looked at viremia. This means HIV load, that you have virus in the blood. We can determine the viral load. Is there any other thing we can do to look for viral load? Let her answer first. You already tried your luck 3 times, I know that. Yeah. All right, go ahead. Now you can speak. She's silent. Yeah. Question is this way, the answer is that way. Yeah, now you can say it again. Biopsy of what? Nothing to do with tissue, nothing to do with colon. Yeah, Ashley.
Or liver.
Nothing to do with liver either. Okay, don't say anything else now. We have to give a reason. Yeah. No maybes. It's to be definite. Say it again. I didn't. Yeah. So what did you want to do? Everybody knows in the cell. Yeah. So blood. Viremia means virus in the blood. What else you want to do? She said tissue. I said no. Say it again. Saliva. Secretion. Saliva, semen, vaginal secretion, where the virus load will be. I just told you this is how it's transmitted. So you can give 1 point to him. Saliva, good answer. So other than virus, because you can also get through transmission of virus via blood transplant, blood transfusion. But these patients are shedding off viral load in all secretions like saliva, but little. The maximum dose, I'm going to talk about that right now, is in the blood, semen, right? And then you can argue if it's semen, it can be also in urine, but you're talking about the viral load. Vaginal secretions, right? And, well, some of them are, I'm going to leave it on you, and I'll just give you the data. Some are, what you say, not proven, so to speak, but we can discuss that later. But that's important. So you can see from here, in terms of primary infection, the virus load, you can see from, let's concentrate on the CD4. So you can say CD4 basically will go over here, and CD4 T cells are in lymphoid organs, and you can see from here the CD4 count will go down. So you can see CD4 count going down till you have less than 200. There's like a death license. And then again, in terms of the symptoms, you will see all the way from acute HIV syndrome to wide dissemination of virus, seeding of lymphoid nodes, and then you can see that you have constitutional symptoms, opportunistic and death, right? The most important thing you keep in mind is that clinical latency is you may not show any symptom over weeks and years. And I said the current information is about 10 years. And if you remember about that woman I said was negative for HIV, she also became positive within 8 years. So that's what latency. So the virus, for some reason, I mean, there are a lot of research, I don't want to go in detail, but just keep in mind that virus goes into the body and disappears and comes back again. Okay, that's called viral latency. Yeah.
So you said that was negative for 8 years, but it was not.
Of course.
But there is no any virus in the saliva or
No. Remember what I said was you can be positive for HIV without showing AIDS. AIDS is a disease. HIV is an infection. Okay, so she was HIV positive, but not showing AIDS. The difference between these two. So you may be infected and not have disease. So what I'm trying to say is that these people have HIV, but that HIV is under control by the immune system for 10 years. Not that they are not passing it on to other people, they are. Like any other viruses. That make sense? Okay, hold on. I need to finish it up, otherwise I'll be distracted. So we talked about plasma viremia, we talked about blood CD4, and then we talked about all these things. You can read it on your, whatever I just spoke about is written for your information. Now, in terms of the test immune response, now that's where I want you to know those antigens over here. So you can see from here, again, these are some of the tests that can be done immediately within weeks. So you can see from here that we are looking at some very specific, for example, these are the viral particles. So you can see if you trace the viral particles are very high in weeks, within weeks, and then tend to decline. And then, as I just told you, can also rise up later. Okay, so there's a trend over here. So that's the viral particle. For a diagnostic test, you can look for viral particles in the blood or in different body fluids. When you look at the CTL specific, which is CD8 cells, so they kind of rise and they plateau, but basically we talk of CD4 T cells. So for this part of course, I'm not going to bombard you with confusion in terms of what's the role for CD8 T cell, but you got an idea that normally you have to have a good CD8 response to fight any viral infection, and this virus goes on the other side to kill the CD4. But CDF specific for HIV peptides are there. As far as antibody is, that there could be an antibody you can test later part as anti-envelope antibody. That's number one. The second antibody you can test was anti-P24 antibody, which was there in the matrix. If you remember, we talked about that. Some of the antibodies which are matrix protein, some of the envelope protein, some of the capsid protein. So this basically gives you an idea as to what is a T cell response and what is a antibody response in terms of what is going on and how we assess the progression of the therapy and how we assess the disease. Now, if you look at the HIV infection, again, you can see over here, these are the CD4 T cell count. They go and drop. So you have seroconversion mean you are exposed, you're positive. That's why we want everybody, regardless of whatever situation we are in, whether we like it or not, we should get for HIV tested. That's the public information for everyone, right? So we want to find out we are positive or not. And then again, we can be asymptomatic or we may have, some of the symptoms like AIDS. The other thing is viremia. Again, you can see viremia is very high initial month and kind of static, and then again comes back in terms of the load. Of all the viremia, if you look at P24 antigenemia, so P24 is a very specific antigen on inside of virus. So we can also detect that. So these brown blocks over here show you that they are here. Most of the time, these people present as persistent generalized lymphadenopathy, persistent generalized lymphadenopathy, which is like a classical symptom of the AIDS. Now, I'm going to leave it on you to read key facts. And we already discussed that. This is just a repeat slide. And again, I just want you to be updated on that in terms of what the CDC recommends. And you can see that these are the body fluids which can transmit through one person to another. We already talked about blood. We talked about semen. We got pre-seminal rectal fluid, vaginal fluid, and breast milk. So these are all considered as the possible sources of transmission of HIV. Okay. Okay, let's move on. And then again, as far as transmission is concerned, I just downloaded it yesterday from CDC website. So this is what they think is a high-risk behavior. And I also told you what is the reason that we don't have that only in a particular group as a typical homosexual, but we have that as a heterosexual in terms of, so what do you think? Why is it that we started off with, this typical classical 1981 description of young white female homosexuals. And now at this level, we are talking about women and heterosexual being the #1 target, even more than MSM. What do you think could be the reason for that? Yeah. Reason. That's what I'm telling you. What's the reason? Yeah. Sexual partners. You're taking more sexual partner, having more? That's what you're saying? Yeah, that's, I mean, so what he's saying is that it is bisexual population. Well, partly yes, but there has to be another reason for that. There's a pathological reason for that. And this is a common sense. I mean, it's nothing special. Yeah. Yeah. Igna. Yeah, that's for all STD. Very specifically, my question is, why is it that we came all the way from one population group to this day when we have heterosexual women in heterosexual relationship as #1 in terms of the incidence of HIV disease? Just hold on. You only that those people take turn who haven't spoken so far. Yeah.
So is it because there are more homosexuals that's having sex with heterosexuals?
That's what he said, and I agreed. The part of that is that homosexuals are bisexual, and they have passing it on to the silent partners of their wives' spouses. Part of it is correct, but there has to be another reason for that. Yeah. Drug use, nothing major. Okay, who else has an answer that? Okay. Yeah, Vivian, yeah. Say it again? Yeah, that's a journal thing. That has happened. I didn't pathologically, yeah. That is partly true, but it was there to begin with. Why is it, okay, if I take that, why is it that incidence is higher in women? Yeah.
Are they just not getting tested for it?
Well, they actually get more tested because they get pregnant and they get tested for it. You only took your turn. Anybody who hasn't taken his turn, her turn? Yeah, you already did. You don't cheat. Okay. Yeah. Anybody else? You compare it with male, right? Yeah. Louder. Partly correct. I'll give you one point. Partly correct because what I said earlier is, in terms of exposure of mucous membrane that is used for sexual activity, she has more. Because had it been male homosexual, they're going to go for a rectal route. As far as female, they're going to have both vaginal as well as rectal. So that increases their chances of getting, I mean, this is a fact. That's what she's saying, very true. Anatomical reason of exposure of a wider mucosa, #1, and being involved in a relationship that may involve both vaginal, and actually, as a matter of fact, 3, vaginal, rectal, and oral. So all these things will stand her out in terms of having higher incidence of sexual activity that happens. So, did I give you a point earlier? Okay, no. Well, you say yes. She did not. Okay. Now, rest of the slide is something that I'm going to let you read on your own. And one thing that I would, again, you can read it on your own. You pretty much talked about that. And one last slide is that if you were to look at HIV in body fluids, there's a lot of research done. But you can see very high in blood. Next is semen. Well, but actually, this has switched no other way. I'll agree with her. No, it's vaginal fluids have more than semen, and even in amniotic fluid. As far as saliva is concerned, again, keep that in mind that when we talk of saliva, for example, what they are talking about is that if it's like a social kissing, it may not, but if it's like an open mouth, deep type of French kissing, of course, you're going to pass it on to the people. That's what CDC thinks. Tattooing, for sure. Tattooing for sure has been quoted at HIV higher incident. Body piercing for sure has been considered, especially if you are piercing those areas which are routinely used for sexual activity in terms of body, because you are opening up a door for all those viruses and bacteria. So we consider that. As far as unsafe injection, again, very important, especially the way we deal. And then again, I'm going to stop here and take any question that you